Alright guys, welcome back to Underground Science, and today we're going to be discussing, or we're going to be picking up from where we left off in the electron transport chain part 1 video. Alright, so remember we left off talking about the complex 1, or also called the NADH dehydrogenase coenzyme Q oxyreductase complex, right? So let's go and get started. So first let's go down here and kind of refresh our memory of what happened. Just a quick overview. So remember that we had our NADH dehydrogenase complex here, complex 1. NADH got oxidized into NAD+, and from this reaction we made hydrogen protons, right? We lost some hydrogen protons from NADH, and there's many hydrogen protons in this vicinity in the matrix, and we'll see how that comes in use later, right now, right? So then what happened is that when NADH got oxidized, it actually lost 2 electrons. All right, these 2 electrons were picked up by flavin mononucleotide molecule. Flavin mononucleotide got reduced to flavin mononucleotide H2. Then what happened is that flavin mononucleotide H2 gave those 2 electrons to iron sulfur clusters inside the NADH dehydrogenase complex. Everything here is happening inside this complex 1. Then the 2 electrons that were in the iron sulfur clusters were then finally transferred over to coenzyme Q, which is not made of protein. All right, and it's a mobile coenzyme. So coenzyme Q again is not made of protein. All right, so let's see what happens next. And then, oh yeah, and we also said when the NADH dehydrogenase complex, when they gave up their 2 electrons, right? When it gave up the 2 electrons, it released energy. And we said that when it did release its energy, 4 hydrogen ions, or 4 hydrogen cations, I should say, were pumped from a pore that's present in the NADH dehydrogenase complex from the matrix to the inner membrane space, all right? And all these hydrogen cations, we'll see many hydrogen cations getting pumped throughout the electron transport chain into the intermembrane space, and we'll see that's important when we get to a future video when we talk about the ATP synthase and how that's the rotor and all that stuff and how that makes ATP. All right, so now let's go and talk about complex 2. So let's go and draw complex 2 up here. All right, so let's go and estimate complex 2. And I guess let's just draw it, just like a regular protein, right? And let's go and color it in, try to at least. So complex 2 is going to be called, let's start out by the name. Okay, so complex 2, it turns out, is called succinate dehydrogenase, right? Succinate dehydrogenase, coenzyme Q, oxyreductase, all right? Succinate dehydrogenase, coenzyme Q, oxyreductase, also known as complex 2. All right, so complex 2 is, remember complex 2, remember succinate dehydrogenase, guys, from the Krebs cycle that we said it was also bound on the cristae? It was the only enzyme in the Krebs cycle that we saw was bound on the cristae because it's involved in the electron transport chain also, right? So let's see how it's involved. So remember in the Krebs cycle we had, we had succinate going to fumarate, right? And then succinate dehydrogenase enzyme was catalyzing that step, right? And then we made 2FADH2 molecules, right? But what happened was that our succinate got oxidized. It lost 2 hydrogens, right? It lost 2 hydrogens and 2 electrons. So here's what happens. So remember, so in the Krebs cycle, so we're just a little continuation down here real quick. In the Krebs cycle, right, which is happening in the mitochondrial matrix, but the succinate dehydrogenase enzyme was bound to the cristae. What happened was we went from succinate to fumarate, right? Remember that from our Krebs cycle playlist or our Krebs cycle videos, to fumarate. And remember that in this step, succinate dehydrogenase was the enzyme that was catalyzing this step, succinate dehydrogenase. And if you remember, succinate dehydrogenase was then bound to the cristae. We're not going to write that here, but it's bound to the cristae. All right, and so also we made 2FADH2 molecules, right? But if we take into consideration of one acetylcoenzyme A molecule, we only make one, right? One FADH2 molecule for each acetylcoenzyme A molecule, right? So So let's go and write that down. So let's go and say we went from FAD, got reduced, and turned into FADH2. Now, if FAD got reduced, something had to get oxidized, and that was succinate. That's why it turned into fumarate, all right? And that means that fumarate lost two hydrogens, but that also means because each hydrogen atom has one electron, we also lost, succinate lost two electrons, all right? So we lost two electrons. So that's why, let's go and see what happens here now. So we see that in the electron transport chain, the opposite reaction happens in terms of FAD and FADH2. FADH2 is actually converted to FAD. All right, so let's go and write that down. So over here, right, FADH2, so FADH2 is then converted to FAD. All right, and when we do that, we get oxidized. FADH2 gets oxidized. And then these two electrons that we lost, right, are the two electrons that FADH2 gets oxidized from. All right, so it's kind of weird. Sorry, what I worded it as, but when FADH2 gets oxidized, it loses two electrons because that's what oxidation reaction means. Something gets oxidized, you lose electrons. FADH2, see, it lost two hydrogens. That's why it turned into FAD. Remember, with each hydrogen, we have one electron. If we lose two hydrogens, we lose two electrons. So then two electrons are lost, right? And it goes into the succinate dehydrogenase complex. Now what happens next is that we give these two electrons over to our iron sulfur clusters. All right, so we give these two electrons over to our iron sulfur clusters inside complex 2, and then these iron sulfur clusters then give these two electrons to coenzyme Q. Now the weird thing is that when succinate dehydrogenase complex or complex 2 gives up these two electrons, it loses energy or energy release, but complex 2 does not pump any protons out to the intermembrane space. So it does not, so no protons are pumped out. All right, so no protons are pumped out from complex 2. All right, guys, so that's the end of complex 2, and in the next video we'll see how we'll see the fact that coenzyme Q will then transfer these electrons to our next complex called complex 3, or also called coenzyme Q cytochrome C oxyreductase. All right, and we'll see how complex 3 works after that. So see you guys in the next video, and can't wait to see you guys in the next video. Hope you guys like, subscribe, and hit that notification bell. All right, so you know when I publish my future videos. All right, see you guys later.